Hello everyone. So now we are going to move on to the fourth themes of the biotechnology principle and then process. So still now we learned about that we are going to connect the DNAs and then vectors with the help of a restriction enzyme and then with the help of DNA ligase. So still now we learned about the some of the tools which is important in the recombinant DNA technology. So now we are going to move on to the last tools which is used in the recombinant DNA technology, and then we are going to find out the process of a recombinant DNA technology. So how should be the process, on which order we should use the process. So that we are going to study in this schemes. So now we are going to move on to the prerequisite. So coming to the prerequisite, the first one is a host. So what is a host? So host is nothing but the place where a foreign DNA or a foreign animal can able to live. So let us take an example of a dog and then lice. So in this, for this lice, this dog going to be a host. Likewise, in a recombinant DNA technology, that vectors, when they are going to introduce their recombinant DNA into a particular cell, that cell become a host. So the host is nothing but the place where a recombinant DNA or a foreign particles can able to live, and then they can able to make their progeny. The next word is a cell lysis. So what is the cell lysis? The rupture of a cell wall. So the cell wall going to be having a many pores, and then they are going to be degrade. So that going to be called as a cell lysis. So when a cell, they are going to be named as a cell lysis, means the cell going to be, cell wall going to be degraded, and then the inner particles they are going to be come out. So that we are going to be called as a cell lysis. The next one is a competent. So what is the competent means? The capability or a efficient to take a particular cell, which to take a particular action, which is called as a competent cell. So in the recombinant DNA technology, when a host which can able to have a capability to uptake, or they are going to accept a foreign DNA, that is called as a competent cell or a competent host. So in this you can see this is a normal bacterium. So when you are going to treat with the calcium chloride, this cell they are going to be ready to accept the foreign DNA. So that plasmid they are going to bound, and then they are going to be introduced inside the cell. So they become a transformed cell. So when a cell they are going to be ready, and then they are going to ready to accept a foreign DNA or anything which is to accept inside, which is called as a competent cell. So the next one is a hydrophilic and then hydrophobic. So what is a hydrophilic and then hydrophobic means? Hydro means water, philic means loving. So when a particular molecule which is going to accept the water, or when they are going to easily merge with the water, or when which is going to love the water molecules, means they are going to be called as a hydrophilic. But whereas when it's going to be hydrophobic means, phobic means fear. So when a particular molecule that they cannot able to accept the water molecules, or they cannot able to easily merge with the water molecules, means they are going to be called as a hydrophobic. So hydrophilic means they can be easily merged with the water, they can easily transport in the water medium. But whereas hydrophobic means they cannot able to pass on in the water medium, or they got a fear with the water molecules to mingle with it. So this is a hydrophilic and then hydrophobic. So the prerequisite is going to be continue. So the next one is a thermostable. So what is a thermostable means? The particular organism or a particular microbes, they can able to live in an extreme heat condition. They are going to be called as a thermostable or a thermobacteria. So thermo means heat, stable means they can able to stable live in the extreme heat. So they are going to be called as a thermostable. So thermostable or thermophilic or bacteria means they are going to live in a extreme heat condition. The next one is a bioreactor. So bioreactor is nothing but a vessel which is used to produce a large amount of a product. So that a vessel which is going to be called as a bioreactor. The next one is a annealing. Annealing is nothing but the binding of a primers or a binding of a probe they become a double strand. So when a single stranded DNA, they become a double stranded DNA means they start to become a double stranded DNA, which is called as a annealing. Annealing means single strand they are going to become a double strand, or the formation of a double strand, which is called as a annealing. The next one is a pathogen. Pathogen means the microbes which can able to cause a disease, they are going to be called as a pathogens. So like a fungus, virus, and then bacteria, they can able to cause a disease means that microbes, they are going to be called as a pathogenic bacteria or the pathogenic microbes. So now we are move on to the objectives. So in an objectives, we are going to study about the process of recombinant DNA technology and then DNA isolation process, and then we are going to study about what is a bioreactor. So what is a bioreactor? What is the use of a bioreactor in the recombinant DNA technology? So this is a bioreactor structure. So there will be an input and then there are going to be a output or a waste. So we will be studying later. So the next tool in the recombinant DNA technology is a compete and host. So first we deal with the restriction enzyme. So we are going to use this restriction enzyme to cut the DNAs, and then once we cut the DNA, they are going to be introduced into the cloning vector, and then once it is become a recombinant DNA, we are going to find out the capable host. So in a compete and host, so that is for a transformation with the recombinant DNA. So DNA is a hydrophilic molecule. So what is that hydrophilic? Water loving molecules. So they cannot able to easily cannot pass on through the cell membrane. Why? Because our cell membrane is made up of a hydrophilic head and then hydrophobic tail. So if it is a water molecule or a hydrophilic molecule, they cannot easily pass on through this hydrophobic tail. So that is why the DNAs are any hydrophilic molecules which cannot able to come out of a cell membrane, or they cannot able to go inside the cell membrane. So for that we need to find a solution to introduce or something, or to remove something from a cell membrane or a cell wall. So in order to force a bacteria to take up the plasmid, the bacterial cell must be first made to be a competent. So first that cell to be ready to accept a plasmid or to take a DNA. So now let us see how we can able to make a particular cell to become a competent cell. So let us take a bacterial cell. So we are going to treat with the divalent cation. So in the divalent cation, for an example, we are using a calcium. So when we are going to use the calcium, they are going to increase the efficiency with which DNA enters the bacterium through the pores in its cell wall. So when we are going to treat a cell with the help of a divalent cation or a calcium, so this they are going to make a pores in their cell wall. So once they are going to make a pores in the cell wall, so that plasmids or a DNA can be easily entered through the cell, pores in the cell wall. So we are going to incubate this cell with the recombinant DNA in a ice. So for a particular time we are going to keep it in a ice. Then we are going to place this in a 42 degrees Celsius. So we are going to give a heat shock. So we are going to sudden increase in the temperature. Then we are going to again put it in the ice. So when we are doing like this means we are going to able to make the bacterial cell to take up the plasmid DNA, and then that DNA can be easily remain inside the cell, and then once we are going to put back in the heat shock, and then again when we are going to put in ice, that cell wall going to be sealed back. So this enables the bacteria to take up the recombinant DNA. So when we are going to place it in ice for a particular medium along with the calcium, then we are going to go for a heat shock. So at that time their plasmid is going to be taken inside. Then after when we are going to put it in the ice, they are going to reform their cell membrane so that the cell can be become a host cell. So this is a one way we can able to make a one cell to become a competent host. So some of the other methods we can use to make a particular cell to become a competent host means, one is the micro injection method. So in this micro injection method, so under a microscope we are going to inject a plasmid or a recombinant DNA. Through the help of a micro injection we are going to introduce in the nucleus. So we are going to directly introduce inside the nucleus. So for that we are using a micro injection. Micro means very small, which cannot be seen with the naked eye. So then injection, so that much very small micro injection we are going to use here to introduce a plasmid or a recombinant DNA inside the cell. The next way is we can able to use a biolistic or a gene gun. So what is a gene gun means? So we are going to have a gun like setup, so which is used to propel the gene inside the plant cell. So in this mostly the plant cell is very tough to break their cell wall. So for that we are using a gene gun technology. So in this what we are going to do means, we are going to make a recombinant DNA coated with a gold or a tungsten. So once we are going to coat it with a gold or a tungsten, we can able to put that in a gene gun setup. So once we are going to put it in a gene gun setup, so when we are going to press the trigger, so the molecule, the DNA molecule, they are going to pass on through the trigger holes in the gene gun. So once they are going to become reach the holes, that gold or tungsten has been captured here, and then they are only the DNA coated molecules only they are going to be put inside the plant cell. So in this we are going to make a gold or a tungsten coated DNA with the help of a bullet. So once we are going to trigger the bullet, so the bullet going to be come here, so that bullet going to be stopped here. Only the gold or a tungsten only they are going to be introduced into the cell with the help of a force which is created by the bullet. So once it is going to be easily penetrate into the cell means that gold or a tungsten can be easily degraded. So only the recombinant DNA can be seen inside the cell. So in this one way we can able to make a cell to accept the plasmid DNAs or a foreign DNAs. So in this we can able to make a normal cell into a competent host. So in this some of the method is like disarmed pathogens. So we can able to disarm the pathogens, and then we can able to use as a vectors, so which is used to allow the infect the cell and then transfer the recombinant DNA into the host cell. So by using a pathogens also we can able to use to deliver the recombinant DNA into the host cell. So these are the some of the methods we can able to make a normal cell to become a competent host. So the next one is the process of recombinant DNA technology. So how the process going to be start and then going to be end. So first one is the isolation of a DNA. So we need to isolate a DNA, and then we are going to fragmentation of a DNA by the restriction endonuclease. Then we are going to isolate a desired DNA fragment, and then we are going to ligate with the DNA fragment into the vector. Then we are going to transfer the recombinant DNA into the host cell. Once the host cell which is going to be recombinant, they takes the recombinant, we are going to culturing the host cell in a large scale. So when the cell is going to be produced in the large scale, we can able to get the desired product. So we will be studying each step detailedly in this upcoming slides. So the first one will be a isolation of a genetic material. Isolation of a genetic material. So always the genetic material or a DNA will be seen inside the nucleus, inside the cell, and then inside the cell they will be seen inside the nucleus. First we need to break the cell wall. So for breaking the cell wall we are using enzymes to break the cell walls. So we are using a lysozyme enzyme for a bacterial cell, and then we are using a cellulase enzyme for a plant cell, then chitinase enzyme for a fungus cell. So by this enzyme we can able to break the cell wall. What happens if the cell wall breaks? They are going to release out the inner particles. So they are going to release out the inner particles. So in our cell, in our cells or a microbe cell, there will be a many components like polysaccharides, lipids, RNAs, DNAs, proteins. So there will be a many number of components. So our desired component is a DNA only. So we are using enzymes to degrade all the materials, the waste particles which is came out from the cell. So in our DNA there has a histones. So you know that DNA will be packed with the help of a histones. So for that we are using a protease enzyme. So this histones is a protein. So this protease enzyme they can able to degrade the proteins. So they are going to degrade the proteins which is present inside the cell, and then which is going to degrade the histones. So once it is going to degrade the histones means we are only getting a desired DNA only. So the pure form of a DNA we can able to take. Then we can able to degrade the RNA with the help of a ribonuclease, and then we can degrade the other materials. Now, so during that process, when we are adding a chilled ethanol to the tube, this makes the DNA to be precipitated out. So they are going to be purified DNA going to be precipitated out. So this is how the DNA precipitate will be seen. So this is a DNA. This is a pure form of a DNA. So now this DNA has to be taken out from the test tube. So for that we are using a process which is called as a spooling process. So in this spooling process we are going to remove the DNA thread from a test tube. So always the DNA going to be precipitate in a form of a thread only, colorless thread. So by adding a cold ethanol we can able to precipitate the pure form of a DNA, then we can able to remove the spooling process. So now we get a desired DNA. Next we want to cut the DNA at a specific location. So for that we are using a restriction enzyme. So we are going to incubate the purified DNA molecule with the restriction enzyme. Incubate means we are keeping for some time. So we are going to incubating a purified DNA molecule with the restriction enzyme. So what happens? This restriction enzymes they are going to cut the DNAs, and then they are going to make the seal in the DNAs. So they are going to cut the DNAs and then they are going to join the recombinants. So first one they are going to cut the DNAs, they are going to act inside the DNA, they are going to cut, and then they are going to make a DNA fragments. So this DNA fragments can be separated with the help of a gel electrophoresis. So now this cell has a purified DNA. Now this incubation cell has a purified DNA, and then with the restriction enzyme we are going to cut the DNAs into pieces, and then these pieces can be segregated according to their size with the help of a gel electrophoresis. So in this gel electrophoresis we can see the end fragments which is done by the restriction enzymes. Then with the help of a DNA ligase we can able to seal the gaps which is seen in the DNAs, or we can able to seal with the plasmids. So next we are going to move on to the amplification of a gene of interest using a PCR machine. So we are going to using a PCR machine to amplify the gene of interest. So we are going to amplificate. Amplificate means multiplication of a gene of interest. So through the gel electrophoresis, the desired DNA can be taken out. So once it is going to be taken out, we need to have a more number of gene of interest. Then only we can able to achieve the product. So for that we are using a mechanism which is called as a polymerase chain reaction. That is a PCR, polymerase chain reaction. So in this polymerase chain reaction, there are going to be a continuous cycle of a DNA amplification. So in a one cycle, there are going to be a three different steps. The first one will be called as a denaturation. So in this process, the temperature would be 94 to 98 degrees Celsius. What happens means in this temperature, the double stranded DNA, they are going to be divided into a two single strand. Then next one is a annealing process. So annealing process which is going to be happen at the 50 to 68 degrees Celsius. So what is an annealing? So this single stranded they are going to become a double strand. The formation of a double strand they are going to start here. How means we are going to introduce a primers. So this primers means which is a known sequence. So this sequence only they can be amplified inside the PCRs. So only this sequence only going to be produced more in the PCR cycle. So they are going to be joined with the DNAs. Then we are going to add a nucleotides. Nucleotides, then help of a polymerase enzymes, they are going to be start adding the nucleotides, and then this single strand they become a double strand. So in the heating process, we are going to separate the double stranded DNA into a two single strand. Then with the help of a polymerase enzyme, that is a Taq polymerase, we are going to make a single strand to become a double strand. So what is this Taq polymerase means? Which is going to be isolated from a bacterium that is called as a Thermus aquaticus. So thermus means heat. So this bacteria going to be live in an extreme heat condition. So why we are using this Taq polymerase means during this process we are going to suddenly increase the temperature, that is from 68 to we are going to increase into 72 degrees Celsius. So for that time, and the normal polymer is going to be easily degraded. So to withstand this temperature, we are using a Taq polymerase. So this polymerase can able to act on the DNA, and then they can able to produce a double stranded DNA. So that is why we are using a Taq polymerase. So this is going to be isolated from a bacterium that is called as a Thermus aquaticus. So Thermus aquaticus means they are going to live in an extreme temperature. So this is how the process will be. So first kindly focus on the temperature, and then kindly focus on the DNA strand. So at a 94 degrees Celsius they are going to divide it into two, and then at the time of a 55 they are going to join, and then at the time of a 72 they are going to elongate. So now this two strand become a four single strand. So what happens in the 94 they are going to again divide it into two, and then they are going to be at the 55 they are going to be add a primer, and then they are going to at the 72 they are going to form a again double strand. So likewise they are going to be a continuous cycling process. So this three steps, when they are going to become in a one single cycle, in a one single step means they are going to be called as a one cycle. So can you see this is a only one DNA strand, that's a double strand. So at 94 they are going to be divided. Then at the 55 they are going to add a primer. So then at the 72 they are going to elongate, and then they are going to produce a double strand. So now this one single strand they have become a two. So now again they are going to be divided, and then they are going to add a primer, and then they are going to be become a double stranded. So initially we have a only one double stranded DNA. Now they become a four double stranded DNAs. So likewise each cycle they are going to produce a more number of copies. Now we are going to move on to the next step, that is the insertion of a recombinant DNA into the host cell or a organism. So we are going to introduce into the host cell. So we are going to introduce the recombinant DNA into the host cell. So we are using a plasmid. So we are cloning vectors. So we are using a recombinant plasmid. So when we are going to introduce into the E. coli cell. Let us take our example of a E. coli cell. So when we are going to introduce into the E. coli cell, we are going to plate it in a medium. So with the help of a restriction antibiotic resistant gene, we can able to find out the E. coli cell which taken the recombinant DNA or not. So let us take if we are going to introduce the foreign gene in a ampicillin resistant gene, means they cannot able to grow in a ampicillin medium. So likewise we can able to segregate. So when we are going to plate it in a nutrient medium which contain ampicillin, if a colony of a bacteria with resistance to the ampicillin, they can contain a plasmid. If we are going to introduce the recombinant DNA in a tetracycline region. So when we are going to introduce a foreign DNA in any antibiotic resistant gene, that gene going to be get inactivated. So with the help of the other antibiotic resistant gene, we can able to find out. So when we are going to introduce in ampicillin, means we can able to find with the tetracycline resistant gene. So when we are going to introduce in a tetracycline, means we can able to use the ampicillin resistant gene. So likewise, So due to ampicillin resistant gene, one is able to select a transformed cell in the presence of a ampicillin medium. So likewise, this ampicillin resistant gene going to be act as a selectable marker, or a tetracycline resistant gene, they are going to be act as a selectable marker. The next one is we are going to obtain the product. So we are still now till now we learned that up to the sealing of a cloning vector, then we are going to introduce into the host cell. So inside the host cell, their ultimate aim is to produce a desirable protein. Let us take a gene which is used to meant for a protein production. So if it is going to be introduced into the host cell, means their ultimate aim is to produce a desirable protein. So the foreign gene get expressed under the appropriate condition only, not in every condition. So after having cloned the gene of interest and the expression of a target protein, one has to consider producing it on a large scale. So still now we are doing in a laboratory. That means we are until the host cell we are going to use it in a laboratory. So once we are going to insert it into the host cell, we are going to introduce this E. coli or any cell, host cell, into a large scale. So then only we can able to produce a large number of products. So if any protein coding gene which is expressed in a heterologous host is called as a recombinant protein, because if it is going to be in a normal cell, they are going to produce a protein, means they are going to be a normal protein. But when we are going to isolate that protein gene and then we are going to introduce into another host cell, in that host cell they are going to produce a protein, means that protein going to be called as a recombinant protein. So grown on a small scale in the laboratory. So in a laboratory we can able to grow only in a small scale. So which is used to extract the desired protein and then purifying it by a different separation technique. So by a different separation technique we can able to purify the proteins or an enzymes or antibiotics or a vaccine, whatever the product will be. So we are using a continuous culture system for the production of a large quantity of a product. So in this continuous culture system, we are going to continuously introduce the cells in the bioreactors or in a medium. So in that medium, they are going to introduce continuously. So what happens means the cell is going to be divided continuously and then product is going to be produced continuously. And then there has a overflow or a outflow. So with the help of this, we can able to collect the microbe cell. So we are going to continuously input, then we are going to get a continuous output. So at the simultaneous time we are going to get a more number of a component. So the concentration of a substrate will be more and then concentration of a product will be more. So that is why they are using a continuous culture system. So we are going to continuously inputting the microbes and then we are going to continuously removing the products. So in the continuous culture system, the growth curve of a microbes will be always remain in a stationary phase. Stationary phase means they are going to be remain continuously in a growing stage. So in that stage we can able to take the microbes out or we can able to take the products. So small volume cultures cannot yield an appropriate quantity of a products. So for that only they are using a bioreactors. So bioreactors are nothing but a large volume of a culture can be processed and it was required nearly 100 to 1000 liters can be do it in a bioreactors and then we can able to get a large quantity of a product. So in a bioreactor, let us take an example of a one product. So now we are going to introduce a input, that is a Aspergillus niger. So we are going to introduce a bacterium Aspergillus niger. So in the bioreactor they are going to grow for a continuous time. Then we are going to remove the waste and then we are going to remove the component. So this Aspergillus niger they are going to produce a citric acid, means that citric acid will be a product. So we can able to get a more number of a citric acid component and then we can able to use for a production of a product. Likewise, whatever the products which we are using now, let us take a shampoos or face creams or whatever it is. So whatever the products means they can be grow in a bioreactors when it is going to be a microbes mediated, like curd or like a bread or something, whatever it is. One example, best example is a curd. So when they are going producing a large quantity of a curd means they are going to be continuously cultured in the bioreactors. So bioreactors are the raw materials are biologically converted into specific products, individual enzymes, etc. using a microbial plant or an animal or a human cells. So they are going to provide a optimum condition for achieving the desired product by providing optimal growth condition. So in this bioreactors, they has equipments. So each one going to maintain a particular temperature, particular pH, particular salt, vitamin, oxygens. So each one going to be controlled in a normal way. So that's only leads to the growth of microbes in a more number. So in that there are two type of a bioreactor. One is a simple stirrer tank bioreactor. Another one is a spage stirrer tank bioreactor. So in these two, they are a cylindrical or with a curved base to facilitate the mixing of a reactor contents. So in the bioreactor, they has a stirrer. Stirrer means which is used to stir the medium, culture medium. So with the help of this stirrer, they are going to mix continuously. They are going to mix the culture continuously. What happens means they are going to provide a proper aeration. If it is going to be a proper aeration, they are going to be a proper microbe growth. So then only we can able to get a more number of products. Then this stirrer facilitate even mixing and then oxygen availability throughout the bioreactors. So this air bubbles, when there is a more air bubble means that can be bubbled through the reactor and then that can be facilitate the oxygen content. So this bioreactors, they can has a pH control, they has a oxygen delivery system, they has a agitator system, they has a foam control system, temperature control system, and then sampling ports. So in this bioreactor, they has each parts which is going to control the each one. So this can be able to control the pressure and then pH, and then we can able to maintain the pH control here. So we can able to control the temperature here. We can able to break the foam, and then we can able to mix the culture broth. So likewise we can able to has each equipment has each role. So they can able to constantly maintain the pH, temperature, oxygen throughout the culture. What is the sampling port means? So in between, if we need to isolate the component or a product means through the sampling port we can able to takes the products. So that region will be called as a sampling port. So small volume of a culture can be withdrawn periodically. So that's why it is called as a sampling port. So this bioreactors, they are going to used in a production of a large quantity of a product. We are moving to the last process, that is we are going to do the downstream processing. So in a downstream processing, the two technique which is a separation and then purification is the most important one. So we need to separate the components and then we need to purify the components. So then only we can go for a market. So a product has to be subjected through a series of process before it is ready for the marketing as a finished product. So before it is to become a finished product, we need to have a several process of separation and then purification. So the product has to be formulated with a suitable preservatives. So then only they can able to keep for a long time. So they need to be formulated with a suitable preservatives. So formulation has to be undergo through clinical trials as in a case of a drugs. So we need to have a more number of a clinical trials as in a case of a drugs. So then only the drugs can be in a pure form and then has a correct formulation of ingredients. Then strict control, quality control testing for each product is also required. So each product should have a good quality control testing. Then downstream processing and then quality control testing, which is going to vary from a product to product. So each product and then they are going to have a different quality control and then different downstream processing. So application of a recombinant DNA technology. So some of the molecular events in a biological process such as cellular differentiation and then aging can also be studied with the rDNA technology. Then making a gene maps with the precision. Precision means nothing but a sequence. So we can able to predict the sequence of a gene maps with the help of a rDNA technology. Then biochemical and then pharmaceutical industry. So making for drugs there comes under biochemical and then pharmaceutical industries. Then production of a transgenic plant. So we'll be studying what is the transgenic plant. So in an upcoming chapters. So for this transgenic plant we can able to produce with the help of a recombinant DNA technology. Then production of a genetically modified microorganism. So with the help of a recombinant DNA technology, we can able to produce a genetically modified microorganisms. So coming to the question part. So once I ask the question and then option, you need to pause the video and then you just think and then you just cross check with the correct answer once you resume the video again. So let us go for a first question. Which one is the true statement regarding the DNA polymerase used in the PCR? It is isolated from a virus, or it remains active at a high temperature, or it is used to ligate introduced DNA in a recipient cell, or it serves as a selectable marker. Answer is B. So once I ask the question, you need to pause the video, then you just think and then you just resume the video and then cross check with your answer. So what is the temperature required for annealing of a DNA molecule? So what is the annealing temperature? 50 to 65, 30 to 35, 40 to 45 or 20 to 25? 50 to 65. So a technique used to make a numerous copies of a specific segment of a DNA quickly and then accurately is translation or a transcription or a light chain reaction or a polymerase chain reaction. So which technique we can use to produce a numerous copies of a specific DNA, our accurate DNA sequence? Polymerase chain reaction. Then introduction of a foreign gene into a plant or an animal cell using a micropipette is dash. Electroporation, chemical mediated gene transfer, microinjection or a particle gun. So which one is used to introduce a foreign gene into the plant or animal cell with the help of a micropipette or a micro needle? Answer is microinjection. Coming to the summary, so in this knowledge imparting session KIMS, we have learned about methods of a gene transfer, isolation of a DNA, cutting of DNA, PCR, bioreactor, and then applications of a recombinant DNA technology. So we learn all this. So okay friends, so with this chapter is done. So this is the last KIMS in this chapter. So we strongly recommend you to repeat listening to all the KIMS as many times as required until you get a complete and then satisfactory understanding of a concept and then confident for appearing any competitive examination. So wish you a great success. Let's meet again in another chapter. Thank you.